So this is Réunion, Mauritius. The main island is Madagascar, and you have here the Comoros with fourth island, and Mayotte, the fourth one, and the Republic of Seychelles, which is composed of several islands. So this is the area where we have several studies in the Indian Ocean, and I will give some, from the literature, some rough idea about the estimated incidence of leptospirosis. But these data should be read carefully because it's very different kind of studies. But I put some sources on the right. There was a recent paper in 2013 that showed that also the third one, the incidence in Mayotte is quite high and is as high as the incidence in Seychelles, which are among the highest in the world, I think. And in Réunion Island, it's 6 for 100,000, which is quite important. In Mauritius, it's very low. There is very few cases reported. It's increasing now. And for Madagascar and Comoros, there is quite no data. This is the human data. For animals, so there are some studies in Madagascar, in bats, in rodents, and also in the Comoros that report the presence of leptospira. So Frédéric Pages, some of you know him from Réunion, just published this paper about the incidence, with this graph showing the incidence between 2004 and 2012 in Réunion Island. So you can see there is a mistake here. It's for 100,000 inhabitants, but it's between 4 and 10. So the average is around 6. And in 2010, they say that it was linked with an increase of rainfall. If we look at the distribution at the district scale, the higher incidence is on the right, on the east of the island. There are more rainfall also, and on the highest part of the island. Okay, so this is something we are working on now. So we are involved in 2 projects in the region. One is called Lepto-I project, and in my team we are working on the spatial analysis. So this project is in Réunion and Seychelles. It was the choice was not to work in nice places, but the fund, we wanted to work in Mayotte, but unfortunately with this European fund it was not possible. So we have another project in Mayotte, which is also a French island, but it's a very different living condition, poor island. And I am coordinating this small project in Mayotte, which is a small project, but we have a lot of interesting things to do there. So I wanted to present about the area where we are working, and I think it's a little bit different than what was said previous day. I'm not a modeler, I'm a geographer, and one of the purposes of my work, I think, is to build some data to help understand the distribution of the disease. But the first problems that we face is regarding I would say, the access to epidemiological data. And we have worked since last year on this first phase data preparation, like for the geocoding of records and aggregation, and how to use anonymized data. So this is a main issue for us now. And so we still are in this phase and haven't started the distribution analysis. And the other aspect, which is very important for me, because I'm a health geographer, is to understand the spatial inequalities of the population regarding health, so regarding health resources, health access, in terms of traveling time, transportation, and the use also, the behaviors. So I will speak a little bit about that later. So for the leptospirosis data coming from the hospital, so I spoke about Frédéric Pagès working at CIR. They manage the database. It's of course confidential data, and the location of patients are known at the address. And the first issue we had was to geocode, to localize, give a XY coordinate to this address. So it was a first work because the addresses are not well recorded, and we had several issues with that. So we developed a form linking to find the right address in the database, because we have a database of the address for the island. And using this form, we could directly localize each patient. So then, of course, they are okay to provide us aggregated data at this district scale, which is okay. For spatial analysis, yeah. Where these cases are reported? Oh, so this is a fictive data. Sorry, it's very important. Just an example to explain what we did, and after 1 year of the project, we're just starting to get the data. So it's lots of discussion. So the second scale, which is fine. It's called Iris in France, and so you can see the difference. And for these both scale, of course, we have demographic and population data. So it's also a good point to analyze. But what we found out is that it was not only in France, but also in Asia, where I worked before. We had difficulties they cannot give us the precise location of the data. And so we had some discussion, and they say, we can give you the streets, but not the number. So sometimes, the streets are very small alone. Or what I get also is some GPS coordinates that was they modified the last digits. So this was not fine for us, and we discussed in our team to propose a simple method, spatial method, to blur the location. So the objective was to have a tool which would be very easy to use, free, that we can provide this tool to the people responsible of the data, and they can give us back some blurred data. So the method is very simple. We have the case, like here, 3 examples. We take a radius, let's say 500 meters, and then we have a random selection of 1 point in this radius, and we take a second buffer. We draw a second buffer around these new points with the same radius. So the final buffer, like this, or we know that every point within this buffer is at a distance from 0 to 2r maximum. In this case, 1 kilometer. So it's very simple, but at least when we apply this, we know that from the original points, we have buffers, and we know the original points are between 0 and 1 kilometers inside the buffer. So we developed a plugin in QGIS. You may know this software, and the plugin was just released 2 or 10 days ago. So you can download it if you are interested. And we had also discussion, of course, with the authority, and they think it would be sufficient for them because we cannot go back to the case. So we have more. We try to develop more tools in this. For example, the one problem would be if in the final buffer you have only one house, you will know where is the case. So the next step will be to evaluate the number of houses inside the buffer and then eventually increase the buffer size to reach a suitable number of houses. So this is, if you know QGIS software, the plugin is coming in a vector because it uses vector data. It's called blurring. Not sure about the name in English, if it's okay or not. We had a lot of discussion about that. So this is the tool, and you have some options which are here, but it's very simple. You have your point layer, you choose a radius, and that's it. You press OK, and you can ask to add the radius and XY coordinate. So we test it with people who don't know about QGIS. In a few press button, they can blur the data, and then the idea was for us to access more easily to data, which should be anonymized, and then we are okay to work with that data. Do you have a question on that? So this is an example with 500 meters. What is important for us with this scale, even blur, we can still do some cluster analysis and which would be suitable for case control studies. The problem we will also test now with Frédéric Pagès is because the buffer is not on the original place, does it have an influence on the statistics that we will do? So I still believe that when we characterize in the environment like a peri-urban or forested, near forest area, I think it will not have too much effect. But we will try to test that. Yes? So for that, we didn't think about this because in Iranian people go far. Everybody has car and roads are good. The idea was to develop some tool which would be generic, which can be reused in other places also. But of course, it's still the same problem for leptospirosis, where the people get infected, and for the epidemiological data in Iranian, we have both. They have, I would say, they have the location of the people, and they have surveys, questionnaires, and for some of them, they know the place where they are supposed to get infected. So for this one, we still have the right GPS location, and for the other one, I'm not sure. We can make the spatial analysis around the house, but is this relevant? That's another question. So this is another example with a bigger buffer size. So the second area that we are working on in the access to health resources, and we have different calculations. So first one, we draw the map of the doctors and medical resources, hospitals, doctors, And from this, we draw what we call the isochrone map. This is the traveling time, but using the road network. And there are now some spatial tools, much more advanced, to take into account the slope or some different, to put some weight in the network. So we have this first calculation that I call here recruitment areas. And the idea is to identify the isolated population who have a difficult access to Earth, and to see after if, okay, there is no case, but maybe there is no doctor or difficult access to service. And we link this, which is really spatial geometric analysis, with some surveys. And we started a survey about the behavior of the people regarding Earth when they are sick, where they go, what is the reason of the choice, and all this aspect of Earth's behavior, to understand also the perception of the distance. And then we calculate, for us, we calculate the distance between the interview and the doctor, the real distance, I mean, with the network, and the perception. So it's another aspect of this work that just started in March. So for population exposure and disease ecology, I will speak about another thing which is important in my team is the use of satellite image. In my team, we are locating in satellite receiving station, so we have an easy access to satellite data, and we use remote sensing. And there is two use that we try to have. First is maybe the most evident one is for the disease ecology. But I try also to use remote sensing to understand some social inequalities. And this is an example in Mayotte. It's France, but we have slums also in France. You can see over here. And this is a Pléiade image. It's a satellite that was launched 2 years ago. And it's 60 centimeters. It's very fine resolution. And the idea here is to identify the housing condition. So here it's what I call precarious housing. Just close to this, you have very nice big houses with gardens and swimming pools sometimes, and housing estates and commercial zone. And this is something we can observe in these images. So we had also, of course, a field work with some students to build a typology of housing, and different kind of analysis with this one, where we use, so from the satellite images, some analyze the density of the houses, the heterogeneity in the textural analysis of the image, and other GIS data like the slope, some things like this to identify these precarious housings. Another one here is maybe more simple, like a hotspot analysis based on the size and density of the buildings. Okay, so this is one of the use of the satellite image that we are doing, and here's the same. We are waiting for the data, but we know already that not in this village. This is Mamoudzou. This is the main city. But lower, there are some in these slums there are some leptospirosis cases. So the idea was to link, but we don't have the data yet for that. So I cannot show you any more analysis on this. And for disease ecology, so there is two ideas. First is to characterize the habitats of what could be the host. I used to do this on rodents. And then to identify in other areas similar environmental patterns. What is interesting with remote sensing and for us is that in all these islands, the data is very heterogeneous. And remote sensing is a way for us to build some similar information over the different islands. So this is the CISOE station where we are working, and so we use spot 5 images from the station, which are 2.5 meters in the spatial resolution. And on the right, this is also a Pleiad, like in Mayotte, Pleiad image, which is 60 centimeters. And I will speak about these different spatial resolution, which are very important for different reasons. We will see later. Yeah.
Are these images and resolution data available for the rest of the world?
So this is the this is a good question. CISOE station was built to provide satellite data for every people working here in this area. So every institution research. Unfortunately for Tanzania, it just I think the east. Because this is the limit of the reception. This is the radome is a protection for the wind and the salt, but behind you have a big antenna, and every day the satellite is going over the station, and we receive the images. These images are free. If you are working here, I don't know, you can get images. I'm not sure. No. So from CISOE station, actually, we have spot 5 image, which is optical image, and 2.5 meters resolution, and we have radar images. So you can use whatever the cloud cover, the rain, everything. We have radar sat. This image, it's another, I don't know what will be the future of that. We get this image for free as a way to test the image. When the satellite was launched, the CNES provided us these images to test the satellite. So we get plenty of image in our field, but we don't have for the whole region. And this is the problem also, that spot 5 image can cover all the region. We have image over the region, but for this we have just small areas. So these images are good because we can have build land use data sets which can be comparable in different islands, but this one we can only study a small area, and we cannot extrapolate what will we observe locally. So for the remote sensing, probably you know about remote sensing, and now we use what we call object-oriented classification, which is the most up-to-date method. What is good with this method is we have homogeneous polygons. It's not a pixel-based analysis. So one of our concerns is to detect wetlands and lowlands regarding the leptospirosis study. And then when we have a classification, we look for indicators of the presence of rodents or bacteria. So this is, I don't know if you can see, 2 animals that were sampled in the frame of the leptoi project, because I didn't spoke about this. There is a rodent sampling. And basically, from land use map, what we get is calculation of distances. What is the average distance from this animal, like the shortest distance to the house, for example, here, or to this kind of vegetation. And after we calculate some statistic for each species, and we build like this some environmental data. The second classic method is to build also a buffer around the animal, which is still the same question: which size, what is the movement of the rodent to change from one species to another one, and also according to the season probably. So we try with different buffer size, and in this buffer size we calculate what we call the landscape matrix. For example, this is an homogeneous area with vegetation. We calculate the number of patch. It's like 1, 2, 3 patch, 3 classes, and this is very heterogeneous with some housing, some wet area. This is a wetland, some wet area, and some trees. So this is the buffer analysis and the landscape matrix. It's quite also classic. So what I said before, with this we can have a study over Réunion, over Seychelles. This is the main island, Mahé, on the right. And this is what was finished recently, the land use map of Réunion Island. And we will have similar map over all the island. So Mahé, Mayotte is almost finished. We also work in Comoros. And so this is the what we call the regional study. And we have also this macro study. It was the image before. So this is a wetland where we reported some leptospirosis cases quite regularly. And so it was in parallel of leptoi project, there was a rodent sampling. So this is the wetland area, was a rodent sampling. This is all the points. And this is a dry area. It's kind of, this is near the only place in France where you have an African savanna. and Réunion is interesting because it's a big biodiversity of landscape. And so this is the sampling that was organized in leptoi project by CRVOI, which is a research center for emerging disease. Of course, it's quite classic with live traps, but the protocol was quite different from the other sites, because it's the access is more difficult. So this is the trapping protocol. And we have also some, we need, in remote sensing, we need also to go in the field because we need to identify what we will see on the image. So this was a field work. And I will show you here to understand the spatial resolution. This is the classification of the same area with Pleiad, 60 centimeters, and spot 5. And on this one, it was quite, some vegetation can be identified quite precisely. And what you see here, it's a rough classification. But the problem here, as I said, the image is smaller, so we cannot work on the whole island. So we have some limits. We cannot extrapolate what we observe in one place. Okay, it's quite the same. So we just get the rodent information. We don't have yet the information on leptospirosis, which is investigated by CRVOE, but we will get. And but in the field, we observed some also different. There were two species, Rhatus norvegicus and Rhatus rhatus. And Rhatus rhatus is quite everywhere, but norvegicus was more in the wet, near the wet area. And also in the morphology of rats, it was like two different populations, but I guess there will be some genetics to analyze that. So in terms of needs, because it was also the question that needs, data gaps. So I said already we don't have the leptospirosis data yet, unfortunately, but it takes also a long time to build this environmental information, and we have to take into account, so it's okay we don't have the lepto data yet. And one of the need is to understand the exposure of people, behavior, and occupation, and on that we plan to have a survey next year and to understand what are the relation of the people with their environment and the water. And of course, when we will have finished this, we will go to risk analysis, but in with statistical methods, because we are not a modeler. So that's my question. Would this data be suitable for mathematical modeling? Okay, and just to finish, this is some of my colleagues in this picture, but we also organize some trainings on the use of spatial analysis for health purpose, and we could have this meeting last year with people from the region. It was a good way to meet and start collaboration with people. That's it.